Hey, Sister Mayo. So our group is all on campus, and so I don't know if this is what you wanted us to do, but we're just going to do the Google Hangout and show you how we got the answers to all of our questions. So who wanted to do question one?
What's on your thing right now?
No.
Okay. So let's see. Start off with. I'll just drop the thing here. So it's a big A, little a, big B, little b, big C, little c.
And big E, little e, big A, little a, big B, little big C, little c, big D, little e, or little e, little e.
All right, so with this, right, there's what we're starting out with. The first question is asking about if we get an A, B, B, C, C, D. Mm-hmm. Sorry, you can see it, I hope. Big A, big B, little C, big D, little E, little E. Anyway, so from looking at this, right, if you cross heterozygous, you get 1/4 of the dominant, whatever letter it is.
Yeah.
And then you get half of the heterozygous. Then you get 1/4 of the recessive homozygous. We're going to use that just for the rest of this video pretty much to explain how we get a fourth versus a half versus another fourth. And so, and that's for when you're mixing heterozygous guys.
So that will apply to A, B, and C.
Yep. And so for this, right, this will be our half. For big B, that'll be 1/4. Right? Mm-hmm. And then C, the same thing, so this will be a fourth as well. When it comes to a dominant with a heterozygous, you'll get 50-50 of the dominant, and then dominant and recessive. And then, so for this D, we would just get half each time. And pretty similar thing with the heterozygous and the homozygous recessive. This will end up being half and half of heterozygous and homozygous recessive. And so then this would be half as well. So you just add up the fraction, and it ends up being 1/128.
So I don't know if you want us to show you all the work for everyone, but you haven't told us that we're supposed to yet, so we're not going to do that. So this is how we did all the rest of them. And so we can show you some of the other work when they're tricky, but we can just kind of go through the answers for question one that we got this way.
So then B ended up being, let's see, 256, kind of using the exact same method. For C, it was a little bit tricky.
Try to pull a fast one on us.
Yeah, we got it. So it was asking how we could, the big thing for C was that it was asking what is the chance of getting recessive, a homozygous recessive, but that's not possible because this is the only one that could give a recessive, but this one could not. There'd be no way to inherit recessive one for C. And then D, we got 1/32, also using the same stuff.
Mm-hmm. Cool.
And then for 2, I don't know if you want us to tell you where we found them, but we didn't write down. We just got it through the PowerPoints in the lecture and everything. So for the paired homologous chromosomes, we got they're sometimes called tetrads and bivalents. And then for B, the structure that connects the centromere to the microtubules of the meiotic spindle is called a kinetochore. Kinetochore. Thank you. And then C, we found that a diploid fruit fly has 4 pairs of chromosomes. So a fruit fly cell in prophase 1 has 16 sister chromatids. And then the fruit fly cell in metaphase 2 is going to have just 8 sister chromatids. For D, we found that a test cross is the type of genetic cross that determines if a purple flower has that specific genotype. And then for E, we found that a non-functional allele is sometimes called a null allele. And then a partially functional one is called a hypomorphic. And then, actually, I'm going to ask you two this right now because I want to make sure I didn't mix it up. So Mendel's law of independent assortment is the two traits, or segregation? Is
I
the two
have
traits?
segregation
Okay.
involving two traits.
And then the independent assortment involves a single trait.
Mhm.
Okay. So then those are the fill-in.
Cool. I can do the next one, and I'll do 5 too if you guys want.
Okay.
Yeah.
Well, I guess the next one we have to send in pictures of our stuff
Yeah.
for A and B, right?
I guess we could explain how we did, this was the PT,
Oh, right. Yeah.
I'll let you take it away.
Okay. So, so the question was for C, how many sister chromatids are found per cell in this organism during metaphase 1? So this is before the homologous chromosomes have split up. And so I'm actually going to write this vertically. So metaphase 1, each of the homologous chromosomes has been replicated. So there's going to be 2 T's. This is the chromosome from mom, this is from dad, and these are the sister chromatids with them. And then there's going to be 2 big R's and 2 little R's. And then what's a big Y and a little y? 2 big Y's and 2 little Y's. And then the question was, how many sister chromatids are found? So each of these, each letter is a sister chromatid. So we have 2, 4, 6, 8, 10, 12 sister chromatids. And then how many chromosomes are found per cell in the organism during metaphase 2? So after one side has gone away and one of the homologous chromosomes is left, we're in meiosis 2 now, and chromosomes is just going to be 3. So before the sister chromatids get pulled apart, each cell is going to have 3 chromosomes. Okay. Well, we can. I mean, this is the Google Hangout is supposed to be kind of us working through it, anyways.
Okay.
So I don't think it's like necessarily like a presentation.
Can I ask questions?
Sure.
Explaining it then?
Sure.
So are you done with this?
Yeah, that is question 3.
Now we can move on to question 4. A horse has 32 pairs of chromosomes. 32 chromosomes.
Pairs.
So there's a total of 64 chromosomes then?
Right.
And so there's 32.
Right, because it's like there's like number 1, 2, 3, 4 chromosomes, but there's 2 of each of those.
So then
That's why there ends up being
pairs.
64.
Yeah, that makes sense.
So how many chromosomes are present per horse cell at the conclusion of meiosis 1? So what has happened at the end, by the end of meiosis 1, what has happened?
Two daughter cells, right?
Yeah, 2 haploid daughter cells,
Okay.
and homologous chromosomes have split up. So his 2 X's that he's drawn separate now.
They're apart from each other.
So how many total chromosomes? This doesn't say how many chromosome sets. That's how many total chromosomes. So that is still 32.
Okay.
Because like it says 32 pairs of chromosomes. That I think you could substitute pairs for sets.
Okay.
In
That's
this.
what confuses me
Mhm.
because I didn't know these were interchangeable.
Right. Yeah, that will probably make it easier to recognize it.
Okay, what part are we on?
We're on.
Sister
I think we're on B.
already.
Yeah.
Prophase of.
How many tetrads are present per horse
The tetrad in this case is each of these, while combined with the really big word.
Yeah, while they're joined by their synaptonomal complex.
Yep. So then each one of these sets would count as one unit. So therefore, there's 32. Even though it doubled, there's still one unit as a tetrad. So that's why it's 32 as well. And then how many sister chromatids are present in a horse during prophase 1 of meiosis, in which this kind of goes back to this. We haven't split yet, so we've got 128 individual sister chromatids if we count each little guy.
Yeah, because if each of these is a chromosome and there's 64 of them, 1/2 of it, because it's been duplicated, is a sister chromatid. Right. Okay, so then how many sister chromatids are present per horse cell during metaphase 2 of meiosis?
That would be here. Metaphase 2, yeah. And so this is counting how many sister chromatids. So we had 128, and now they're split. This would be 64 now, because dividing in half.
Right. We haven't split sister chromatids up. We've only split homologous pairs, but still it's 64. And then how many bivalents are present per horse cell during anaphase 2 of meiosis?
So for this one, we said 0 because bivalent is the term used during prophase. Not sure.
Yeah,
maybe on that. But that's what we were thinking.
Yeah, the way we understood bivalent and tetrad was when homologous chromosomes are combined or they're connected at their chiasma. So we have 0. Okay, moving on to question 5. Question 5A is dealing with phenotypes. I don't know if this is how you wanted us to do it. This is what we found was somewhat, I don't know, easier to see and understand. We just separated each of the genes because the genes are inherited independently. So we can, just like we did in the very first question, you can take height by itself and try to figure out the probability of being tall or short. And so if you cross, I guess what are the parent genotypes? So it's big T, little t,
big R, little r,
big R, little r,
little y, little y,
little y, little y,
big A, little a. And then the other parent is big T, big R, little r, big Y, little a, little a.
Okay, so this is almost the same probabilities as the other one.
That's true.
So for the phenotype, you can do your Punnett square and figure out which one of these is how many dominant, how many offspring they're going to be with the dominant gene. And so you're going to have 3 out of the 4 are going to be tall, and 1 out of 4 is going to be short or dwarfed. And so we're using this kind of probability for all of them, except for, I guess, these two. So we can kind of show a Punnett square for that one. So little y crossed with big Y, little y ends up with 1/2. 1/2 is going to be green, 1/2 is going to be yellow. And so I'm going to slide over here. So if we want to kind of make it easier to see, we put 3 out of 4 is tall and 1 out of 4 is short. And then you can do the probabilities of the next ones as well. 3 out of 4 is round. Is that what I mean? Round. And wrinkled. 1 out of 4 is wrinkled. And then
Yellow and green.
Yellow green is 1 out of 2 is yellow, 1 out of 2 is green. And then last one is 1 out of 2 is axial.
Axial, and then terminal.
2. So I'm not going to show you. You could do this for all of it. It kind of is time consuming, but this kind of shows why the fractions work. So what's the question for A?
What proportion of the offspring will be tall round, green, and terminal.
So if we follow this line, we multiply 3/4 for the tall by 3/4 for the round. Then you go by 1/2 for the green, and then 1/2 for the terminal. That ends up being 3/4 times 3/4 times 1/2 times 1/2. And I think we have that being
9/64.
that equals 9/64. So we did that for all of the other things. B is for the genotype, and I guess C is as well. And then D is for the phenotype. But for B, we got 1/32. For C, we got 1/16. And for D, we got 1/64 using the same method.
Yeah. And then, yeah, for the genotype, yeah, it's the same thing we did in the first question.
Right.
Yep.
And that is it. If we did this wrong, let us know, and we're sorry, but this is easier than trying to do an extra Google Hangout when we're all on campus.